Hello everyone, welcome back to Microbiome Solutions. Today we're going to discuss about ELISA test or enzyme linked immunosorbent assay. So ELISA or immunosorbent assay are quantitative immunological procedures in which the antigen and antibody reactions is monitored by enzyme measurements. So the term ELISA was first used by Ingwall and Perma in 1971. The ELISA test or the enzyme immunoassay was the first screening test commonly employed for the identification of HIV virus. And this test has got a great sensitivity. ELISA is the abbreviation of Enzyme Linked Immunosorbent Assay. It is useful and powerful method in estimating nanograms per ml to picograms per ml of materials in the solution. So we can say it is highly sensitive and a specific test. Why it is known as enzyme linked immunosorbent assay? So the antigen of interest is absorbed onto the plastic surface, thus it is called the sorbent. Antigen is recognized by a specific antibody, thus there is an antigen antibody reaction. So the term immuno, this antibody is recognized by a secondary antibody which has an enzyme attached. So enzyme linked. Then the substrate reacts with the enzyme to produce a colored end product. That is why it is called as enzyme linked immunosorbent assay. So the basic principle of ELISA, it uses an enzyme to detect the binding of an antigen and antibody, or it detects the binding of antigen antibody in a solution. So the enzyme converts a colorless substrate, which is commonly called as chromogen, to a colored end product indicating the presence of antigen antibody reaction. So an ELISA can be used to detect either the presence of antigens or antibody in a sample depending on the test which is designed. So if you want to design or if you want to detect an antigen of interest from the patient sample, your test kit would be labeled with an antibody and vice versa. Principle is based on the formation of antigen antibody complex, which is detected by a chromogenic substance or a colored substance using an enzyme conjugated with a secondary antibody. This conjugated enzyme will act on a specific substrate which is called as a chromogenic substrate and generates a colored end product. So this product is qualitatively or quantitatively can be read using an ELISA plate reader. So usually the components of the kit would be pre-coated or stabilized 96 microtiter, well plate, sample diluent, standards and controls, conjugated detection antibody, 10 ml of water solution, substrate and stop solution. So here what you are seeing is a ELISA plate and this are called as the ELISA wells. So each microtiter plate is having a 96 wells. What are the reagents and what function do they perform? First one is antigen. The ELISA plate will be coated with the antigen or antibodies of interest. As I have told before, if you want to detect antigen in a patient sample, your ELISA plate will be coated with antibodies and vice versa. Primary antibody, they are human serum IgG, IgA or it can be IgM. Secondary antibodies will be peroxidase labeled anti-human IgA or IgG or IgM. Antibodies that bind to the antibody complexes. Next is the enzyme substrate 3,3', 5,5' tetramethyl benzidine or TMB substrate, which is a colorless solution that when oxidized by horseradish peroxidase or HRP will in turn turns blue. Next is stop solution to stop the enzyme substrate reaction, that is 0.5 normal sulfuric acid. And the enzymes commonly used in ELISA, horseradish peroxidase, which is the most commonly used enzyme, alkaline phosphatase, beta-galactosidase, lactoperoxidase, and the substrate used is tetramethylbenzidine or TMB. And the specimens or samples for ELISA, it could be serum, CSF, sputum, urine, semen, supernatant of culture, and stool. This is about today's lecture. If you have any queries, please post it in the comment section. Please do like, share, and subscribe to our channel. Thank you.